Alright, hello everyone. So, welcome again to another video of biological science topic. So, in this video that we were going to have this day, we were going to discuss the chemical basis of genes. So, basically, we were going to discover the structure and function of the DNA and the RNA. Alright, so let's get started. Alright, so if you remember, if you try to look a blueprint of a house, alright, so what you see on the blueprint are the design of the house that you are going to build. Alright, so there's no logical reason that there will be no logical reason if the blueprint of the house and the actual house that you will build will not be the same. So the blueprint always, or the actual house that you are going to build always follows the blueprint that you are using. Alright, so what is the connection of this analogy that we have right here? Our body, alright, so our body, the structure of our body follows a certain blueprint, a chemical blueprint, just like what we have here in this house. Alright, so we call that blueprint as DNA. Okay, so DNA stands for deoxyribonucleic acid. So we have it right here. So deoxyribonucleic acid, DNA. Alright, so this is a nucleic acid, so one of the biomolecules that comprises all living things. So this is a nucleic acid that contains genetic instructions used in the development and functioning of all known living organisms. Please take note, all known living organisms. But there are some cases like the viruses, they contain DNA. Alright, so meaning to say that they also have the function, or they do have genetic instructions upon their it contains instruction upon use in order for them to reproduce. Alright, so meaning to say that the DNA is a biomolecule that contains a blueprint in which the structure of an organism, the organs that it will develop, then or anything else about, or everything and everything about that organism will be seen on that particular organism, and that is contained in a that is contained in the nucleic acid called deoxyribonucleic acid. So similar to a blueprint, DNA is also a blueprint which codes for what kind of organism are you, how many body structures do you have, and anything else regarding that particular function. Alright, so that is the DNA. Now, the DNA that we see right here is composed of different entities. So we have carbon, we have oxygen, we have nitrogen here, then we have phosphorus. Alright, so okay, so and also we do not forget the hydrogen. Alright, so the hydrogen, so these are the things that comprises your DNA. Okay, so the one that we have right here. And okay, so the people behind the discovery of this wonderful chemical that we have right here, which is the DNA, is none other than Francis. Alright, so we have here in the picture, we have Francis Crick and James Watson. Alright, so we have Francis Crick and James Watson. And James Watson. Alright, so along with Rosalind Franklin and Maurice Wilkins, they do research on what kind of what will be the structure of this or what will be the structure of the DNA molecule. Alright, so and eventually after doing years of research about this structure, they be able to find out that the structure of the DNA is a double helical. It is made up of a double helical structure. Alright, so the one that we have right here. So it is similar to a ladder, which is when you twist a ladder. When you twist a ladder, it will look like something related to this. Alright, so the DNA is somehow structured as a double helical model that we have right here. And also they did find out as well that the DNA is made up of a four-letter code. Four-letter code that we have right here. We have C, A, T, and G. Alright, so later we will going to discuss which codes or which letter pairs with another code, and so on and so forth. And what will be the effect if this code is somehow translated into something. Alright, so that's the structure of the DNA. Don't forget, it is a double helical structure. So if you twist a ladder, then that's the DNA. Alright, so next one. Alright, so where is the DNA located? So the DNA is located basically on the nucleus. Alright, so this is the cell that we have right here, and the nucleus is located at the center of it. Okay, so now in this case, so inside the nucleus there are another structure that is found there, which is called the chromosome. Then when we enlarge that chromosome, so this chromosome that we have right here, which is a structure that contains all genetic traits and other stuffs related to an organism. Alright, so and also it contains certain numbers. It depends upon the organism that you are studying with. For example, in humans, we do have 46 chromosomes in our nucleus. Not only in one part of the body, but all parts of the body contains this number of chromosomes. If you are a human, and if you are other animals, for example, the fly, it contains a small amount of chromosome, which is 8. Alright, so and basically they look like this. Alright, so we have the centromere over here. Alright, so we have the centromere, and we have the chromatids on both sides. So we have chromatids. Right, so and when we enlarge this chromosome, that we have here, it will look like this one. So it will look like this one. Now the balls that you see here, this thing that you see here, these are proteins. Alright, these are proteins called histone. Alright, so histones. Now if you happen to see, a hair clip, which is used by some women to tie up their hair, so it is basically just like that. So these are proteins that coil the DNA together. So this is the DNA. It coils the DNA together so that it will not be scattered around the cell. All right, so this is one of the basic feature of a eukaryotic cell. So all eukaryotic cells have these histones that makes the DNA packed together and it is packed together and as well organized, so that it will not go anywhere else inside the cell. Now the prokaryotic cell, so the thing that you see here, this histone, are only found in the eukaryotic cell. All right, so the prokaryotic cell doesn't have this doesn't have any proteins like histones that we have right here. So that is why they don't have distinct nucleus, and their chromosomes, like the bacteria, are scattered all throughout or inside the cell. All right, so that is the histone. Now unpacking. Right, so unpacking this particular DNA or this particular chromosome, if you remove the histones, you will go to the structure of the DNA, which is the double helical structure of it. Then if you untwist this double helical structure, you will see the sequence of DNA and the particular gene that the DNA is the gene in which the DNA is located. All right, so this is your nucleus, the chromosome. Then when you uncoil it, you got the double helical structure of the DNA and the sequence of the DNA that is found in every gene inside the body. Alright, so with the discovery of this DNA, it opens the gateway to more researches like cloning, alright, so genetic gene recombination, something like that, alright, so and other stuffs that is related to something like molecular biology. Alright, so it opens the gateway to researches in molecular biology because of the discovery of the structure of the DNA and also its function. Alright, so in this topic that we have right here, so we were going to discuss the parts of the DNA. So if you don't know already the parts of the DNA, so here we go. So the parts of the DNA, one of it is that makes up its name, or the one that makes up its name, which is deoxyribonucleic acid. So one of them is a sugar named deoxyribose. All right. So in this case, or in this particular picture, as you can see, it is made up of 1, 2, 3, 4 oxygen molecules or oxygen atoms, and it contains carbon atoms, 1, 2, 3, 4, 5 carbon atoms, and a lot of hydrogen molecule. And please do take note that the deoxyribose sugar is always shaped like this. All right, so an oxygen on the tip of the pentagon. Alright, so it is always shaped like this. This is a sugar molecule. Alright, next, and deoxyribose sugar is attached through a phosphate group. Alright, so as you can see here, there's a phosphorus on the middle, and 4 oxygens is attached to it. Alright, so this is the phosphate group. Again, this is attached through the deoxyribose sugar. Alright, next one is the one that makes up all the letters in your DNA, the ATCG, which is the A stands for adenine, the G stands for guanine, so guanine, G-U-A-N-I-N-E. Next one is the thymine, which is T, and cytosine is letter C. All right, so these are the nitrogenous bases, and as you can see here, these are pyrimidine and purine bases basically. All right, so okay, so nitrogenous bases, one of the most important parts of your DNA. All right, so when they combine together, they are called nucleotides. So we have phosphate, then we have the ribose sugar, and whatever nitrogenous base that you have. In this case, we have the adenine. Now, this one unit that you have right here is called nucleotide. Alright, so this one unit. Now, when you connect this nucleotide to other nucleotide, we call it polynucleotide. So, in this picture that you see here, you have now the phosphate group, then we have the deoxyribose sugar, and one of the nitrogenous bases, which is connected to another nitrogenous base. Alright, so basically, how many nucleotides that we have right here? 1, 2, 3, 4, and 5 nucleotides. So, meaning to say, group of nucleotides that are attached together is called polynucleotide. So in this DNA, or in this structure that we have right here, this is a polynucleotide structure of the DNA. Alright, so the nitrogenous bases is connected via hydrogen bond that we have right here. And also on the other side of this picture, this side also has 1, 2, 3, 4. It has 1, 2, 3, 4. Yeah, 4 and 5 nucleotides that makes up this polynucleotide molecule of the DNA right here. Alright, so again, the nitrogenous bases are paired together, alright, paired together properly in this particular polynucleotide. Alright, so how do we pair up this? Codes or these letters that these nitrogenous bases that are found in the DNA? There's always a rule. You don't always pair A to G. You don't always pair A to C or something like that. They follow a certain rule that adenine is always paired with thymine. So meaning to say that the A cannot pair with G or C. Alright? And also the T cannot paired with C or G. So meaning to say that adenine is always paired with thymine. So that's the rule of thumb here. So the other one is the cytosine, which is the letter C. It is always paired with guanine. So just like the first one, it cannot paired with A and T. So it is always paired together. That's the rule of thumb here. So that's how you base pair, or that's how you base pair these nitrogenous bases found in DNA. So in this polynucleotide, the one as you notice, they are not paired. They are not paired randomly. They are paired according to this base pairing rule. And this base pairing rule, or this pair that we have right here, that makes these bases a complementary bases. So meaning to say that the A is complementary to T and C is complementary to G. Always. Alright, so that's the rule of thumb here. Next one. Now, Erwin Chargaff, James Watson and Rosalind Franklin, they do research about this particular structure of the DNA, and they arrive at the same conclusion. Their research arrive in the same conclusion, although they have separate researches. Three of them arrive in one certain conclusion. Now, Chargaff used this particular observation to form a conclusion that the DNA from any kind of cell or any cell of all organism should have the ratio of 1:1 pyrimidine and purine bases. All right, so it is always 1:1. If there is 14 adenine in that particular cell, then therefore it always have a 14 thymine on that particular cell. Never, in this particular rule, it says that the 14 adenine and 15 thymine will never or will not exist. So it is always a 1:1 ratio. Alright, so of the pyrimidine and purine bases. So what are these bases? This is the adenine and thymine and the cytosine and guanine. Alright, more specifically, that the amount of guanine is equal to cytosine and the amount of thymine is equal to adenine. Alright, so yan. So this is the purine. So the purines are the adenines, alright, in your nitrogenous bases, and also the guanine. So for you to be for you to have not having difficulty memorizing this one, all the shapes of all the nitrogenous bases that has this shape, these are called purines. And on the other side, we have the pyrimidines. So all pyrimidines have this kind of shape. So the cytosine and the thymine, all right, the cytosine and the thymine have this kind of shape. They are called pyrimidines. Now, in RNA, there is what they call uracil. In RNA, there is what they call uracil that replaces the thymine there, and also it is shaped like this. So if it is shaped like that, it is considered to be pyrimidine. So according to Chargaff, the ratio of these things that we have right here, except for the uracil, so the ratio of the cytosine, thymine, adenine, and guanine in a particular cell always arrive at 1:1 ratio. Alright, so with that, let me give you one example here. For example, in humans, for example, I will just make a hypothetical value of the adenine and thymine. For example, in human, alright, if we took a DNA sample from your liver, for example, the human liver. So, for example, the DNA ratio there is around 9.7. Alright, so 9.7 adenine. So the ratio of adenine and thymine is somehow is 9.6. Alright, thymine. Alright, so in this case, okay, so in this case that we have right here, so the human liver contains this kind of ratio. Now if that is a human, so if we took a stomach on that same organism, the human, so meaning to say that you will have the same number or the same ratio of these nitrogenous bases that you will expect to find in the stomach. So meaning to say, if that is the human, all the ratios of these nitrogenous bases should be equal. So in the stomach, it should be equal. So it might change a little. For example, in the stomach, there is what they call for example, there's 10.2 adenine, and the thymine is 10.3 thymine. So still, it is 1:1 ratio because it belongs to a same organism, so that's the reason why it contains the same ratio, 1:1. It is always 1:1. Now, in other organism, regardless of which, a whale or as small as a bacteria, so as long that it contains this kind of nitrogenous bases, which is arranged in 1:1 ratio, yeah. So it means that it belongs to one particular group of organism, regardless of what organ you took from that particular organism. Alright, so next one. Okay, so we have here one parts or one place in a one place in the DNA called the genes. All right, so the genes, if you recall the lesson in your genetics, the heredity by laws of heredity by Mendel. All right, so the genes that we have right there are the following. So for example, Mendel studied the peas that he used to grow in his backyard. All right, so he studied there how the height what are the height of this particular plant. So there are tall plants and there are short plants. All right, so this is the height. Right, so for you to be able to understand what are genes all about. So another one is the color of the pod. So the color of the pod, some are green and the others are yellow. All right, next one. All right, there are pods that are the shape of the pod. All right, there are pods that are inflated. All right, so there are inflated and there are wrinkled. All right, so sorry, they are others are inflated and others are the opposite of this inflated. All right, so I forgot that one. So let's use another one. I forgot the opposite of this. Yeah, ayon. All right, I remember already. Inflated, the other one is constricted. So meaning to say that the pod you'll be able to see how many seeds that the pod contains, because it is easily seen because the pod is constricted. Unlike the other one, which is inflated, you don't have any idea how many seeds are present inside. So the other one is constricted. So constricted. All right, so what we have right here. Alright, so what do we have right here? These are the genes. These are the traits. Alright, so the thing that we have right here. So if you're going to ask, are these traits that we have right here genes? No, they are not genes. Alright, so the genes are the one that is encircled here, the height, the color, the shape, and other things that Mendel observed on those plants. Alright, so what are genes? These are physical and functional unit of heredity. So what do you mean by physical? So the shape. Alright, physical. So shape. Color, also physical. And the functional unit is meaning to say, since it is physical, whatever description or whatever description that the physical aspect of that gene is, and meaning to say that the functional unit of it will be how it will manifest on that particular organism. For example, the height. Alright, so if it is tall or short or something like that, so its function on the organism. Alright, so the height. Next one, the color. So its function is to provide color to the pods or the seeds, something like that. The shape. Alright, so the function of it is to provide a structure or shape on that particular pod. Alright, so basically this is the physical and the functional unit of heredity. And this particular traits that we have right here is somehow transmitted from one offspring to, or by the parent to its offspring, and the offspring will become a parent and will be transferred to its offspring. Alright, so these genes also is made up of DNA. So that's the reason why the genes are found in the DNA. Alright, so these are segments in the DNA. And it contains instructions. It contains instructions that creates proteins. Alright, so in our next lesson that we have, we will discover how DNA becomes protein. Alright, so is that even possible? Yes, that is possible. Alright, so these genes contains instructions that creates proteins. To these proteins are responsible in creating these particular traits. Now in humans, we have eye color. We have brown, green, we have brown, or we have blue colored eyes, something like that. And also hair color, we have blonde, we have black, we have gray hair, something like that. So these proteins, which are found in which are instructed inside the genes, creates these particular traits that we have right here. Now, what are these tall? Alright, what are these tall and short, green, yellow, inflated, and constricted that are observed by Mendel? These are the forms of the genes. These are called alleles. Alright, so the tall, short, constricted, inflated, blonde, blue eyes, green eyes, red eyes, brown eyes, these are alleles. Alright, so these are alleles. Now, the alleles are found in the same gene. They are found in the same gene. That's the reason why these tall and short are found in height. You don't find tall and short in the color. Alright, so if you get what I mean. So logically speaking, that the tall and short should be found only in the gene that is coding for height. The green and yellow is only found in the gene that codes for color, and so on and so forth. Alright, so these small differences contributes to each person's unique physical feature. Alright, so that makes this particular alleles that we have right here, the one that you have studied before, which is the homozygous one, or the dominant T, the dominant, the recessive T, which is for short, then the capital T is for tall. So these things that we have right here, these are alleles. Now, whatever form, alright, whatever trait that the organism will inherit to his parents, yan, to his or her parents, yan. So these particular physical features that makes an organism a unique organism, that makes an organism varied, alright, group of organism varied, or population varied among each other, alright. So because of these alleles, that creates variation in a population, alright. So that is the genes, alright. So, in this picture that you see here, genes are just segments of DNA that codes for certain features in our body. For example, this segment, for example, that we have right here, example, it codes for the height, or even it codes for the color of the eyes, alright? It codes for the color of the skin, or anything. Alright, any trait that you think of in a particular organism. So that is the gene. Now, located inside this gene that we have right here are the alleles responsible for the uniqueness of the individual. For example, if this is the height, so we have here the tall. For example, we can cut this segment for short, and we can cut this segment for tall again, and so on and so forth. Alright, so this is the gene that we are talking about earlier. Alright, now what is the role of DNA? So we already know its structure. It is made up of deoxyribose sugar, it is made up of phosphate, and it is made up of several nitrogenous bases. Now, what is the role of the DNA? Now, the role of the DNA, it is the long-term storage of information. Information of what? Information of you. Alright, so information of you. Alright, so it means that the DNA, your DNA, contains all the information needed in order to create another you. Right? So if you get what I mean. Okay, so, and also the DNA is needed in order to create the proteins, right, via protein synthesis, which is needed by your body in maintenance, in reproduction, and so on and so forth. Alright, so these proteins are very important in maintaining your body's number of cells, alright, hormones needed, alright, so the neurotransmitters, or anything else, right, from muscles to the smooth muscles of your digestive system, all of those are made up of proteins, and these proteins are coded in your DNA. Alright, next one. The DNA also replicate, alright, so if they replicate, for what purpose? For growth and maintenance of body parts. So that's the reason why our skin, when we take a bath and scrub up our if we scrub our skin, or if we scrub our skin for dead skin matter, yan, so the skin replaces itself with another skin. So in this case, maintenance and growth of body parts is very important in an organism, so that the number of cells is maintained all throughout its lifetime. So replication of the DNA is very important. Alright, so if we take an analogy, or if we use another analogy about DNA, the DNA is just like a hard drive in your computer that contains all the information, all the documents that are found in your computer. Alright, so all the programs, all the necessary things that you need in your computer. Alright, it is found in your hard drive. So it is something like the DNA does. Alright, so next one. and that is the DNA. Right, so that's the function of the DNA. All right, only by death of the particular organism, that particular long-term storage becomes will disappear. All right, all your information, all the traits that you have in your DNA will disappear when you die. All right, so that long-term information will disappear. All right, so next part is the RNA. Now, the RNA is somehow, if the DNA is located in the nucleus, so RNA is somehow located in the cytoplasm. All right, so it only enters the nucleus via instruction or during the protein synthesis. All right, so what is an RNA? An RNA is a ribonucleic acid. So this is ribonucleic acid. So which means from its namesake, it is different completely from your DNA counterpart. Now, biologically speaking, alright, so the RNA is made up of a long chain of nucleotide units. So, meaning to say, based on this description about the RNA, it is somehow different in terms of structure with regard to your DNA. Alright, so basically, the RNA is single-stranded. Alright, so the RNA is single-stranded while the DNA is double-stranded. Alright, based on this description that we have right here. Okay, so next one. So there are different types of RNA. So basically, these types of RNA depends on its function. Alright, so the first RNA that we will have right here is the molecule called mRNA. Yeah, the mRNA is a molecule which is used by DNA in order to create a chemical blueprint. Alright, so meaning to say that the mRNA is the one that copies the DNA blueprint so that the mRNA can code up a protein based on what the mRNA copied on the DNA blueprint. Alright, so as I said earlier that the DNA contains codes that create protein product. Now, the DNA cannot go out of the cell. Alright, so there is what they call an mRNA that goes inside the nucleus. Alright, so the DNA cannot go out of the nucleus, so the mRNA goes into the nucleus to copy whatever blueprint or whatever codes that is stored by the DNA in order to create a protein product. Alright, so that is the mRNA. Another type of RNA is called the tRNA, and this tRNA, from the word itself, or by the way, the mRNA the M stands for messenger RNA. So, from its function, you know already why it is called messenger RNA. Now, the tRNA, or also known as the transfer RNA, from the name itself, it transfers this particular chain. Alright, this particular chain of amino acid that is created using the blueprint in the mRNA, it transferred this particular chain to the ribosome so that it can create protein during a certain process called translation. Alright, so basically from the name itself, it transfers this particular polypeptide chain. So meaning to say, it will create first an amino acid. So a group of amino acid. Alright, so amino acid and another amino acid. This particular chain that we have right here is called the polypeptide chain. Or basically, this is already a protein. So an amino acid, so the protein is made up of amino acid. Alright, so basically, that's how it works. Alright, next one, the transfer RNA will go to a structure that will create the protein which is needed by the body or which is needed by the cell. Alright, so this is now the rRNA, or also known as the ribosomal RNA. Now, this is the part where you create now the proteins that is coded using the DNA blueprint that is copied by the mRNA. Alright, so the protein that is coded here, or the protein that is manufactured here, will be used by the cells in order to, or on its on its activities. Yan, on its different activities, these proteins. Alright, so let me give you one example of proteins. Alright, proteins examples are hormones, something like that. Alright, so another one is the protein found inside the cell. Okay, and this protein that are found in the cell membrane. Right, so another one is the enzymes. Alright, those are proteins as well. So yeah, so basically there are three types of RNA. We have mRNA, tRNA, and the rRNA, and these are their function. Okay, so now these are the people who studied and discovered the presence of this particular nucleic acid. All right, so the nucleic acid was discovered, including the DNA. It was discovered by Friedrich Miescher. All right. So how about James Watson and Francis Crick? Did they discover this nucleic acid? No. They already know that this nucleic acid is present, but they want to know what is the structure. So basically, they discovered the structure of it. Alright? But Friedrich Miescher is the one who discovered the presence of this nucleic acid on all organisms. Alright? So he doesn't know what is this nucleic acid is all about. Alright? What is this nucleic acid is named of. But it is made, or this is what all organisms on Earth is made of. Alright? So next one. Alright, so we have right here Severo Ochoa. Excuse me for that. So this scientist, this fellow that we have right here, discovered the RNA and how it is synthesized. Basically, he discovered how RNA came to be and how RNA is produced inside the cell. With that, he won a Nobel Prize for medicine in that particular research. Alright, so this is the structure of the RNA. So as you can see here, there's some sort of a familiarity in terms of the structure of the RNA. So as you can see here, it is a single-stranded molecule, and it is made up of same it is made up of same components the way your DNA is made up. Alright, so what do we have right here? We have cytosine. Excuse me. Alright, so we have cytosine, and we have the guanine, we have the adenine, and in RNA we don't have we don't have thymine. Instead, it is replaced by uracil. Alright, so these are the four basic nitrogenous bases found in the RNA, and it is not double helical in structure, but it is a single-stranded unit. Alright, so it is a single-stranded unit, and it is a long chain of nucleotide, and that is your RNA. Alright, next one. Its composition. So as you can see here, it is somehow similar to a DNA, but it differs in this particular area. Okay, so that's the composition. This is the ribose sugar. Again, it contains oxygen on this side. So what it differs is on this side. Yeah. So in the DNA, there is CH3OH on this side, but in RNA, it is just OH. Alright, next one. The phosphate group is also present, the phosphate backbone. Next one is the four nitrogenous bases which we encountered earlier. Yan, so we have the adenine, thymine, or sorry, not the thymine one, adenine, guanine, uracil, and cytosine. Alright, so basically the adenine is paired with uracil and the guanine is paired with cytosine. Alright, so next one. So this is what the RNA nucleotide looks like. So it is basically same with the DNA, but with the difference in its sugar, which is ribose, and also some differences in its nitrogenous bases. Basically, this is uracil that we have right here. Alright. So we go now to the summary between the difference between DNA and RNA. So the DNA is made up of deoxyribose sugar, while RNA is made up of a sugar unit called ribose. Now the nitrogenous bases is basically the same, but they do differ only in thymine and uracil. All right. So next one. Another difference is the ratio of A and T. Alright, and C and G is always 1:1, but the RNA doesn't follow that Chargaff's rule. Alright, so why does it follow Chargaff's rule? It's because that the RNA is made up of a single strand molecule. So logically speaking, if you are made of single strand, your nitrogenous bases doesn't have a partner on the other side. Alright, so unlike DNA, it is double helical in structure, so it should be made up of 1:1 ratio of this particular nitrogenous bases. Next one. The DNA is large and insoluble molecule. Meanwhile, the RNA is soluble molecule. Alright, so next one. The DNA is found in the nucleus. Only found in the nucleus. It doesn't go out the nucleus. Meanwhile, the RNA is this is a temporary molecule. This is only created during protein synthesis. All right, so that's the RNA. Okay, and it is found in the cytoplasm, and it only enters the nucleus when it is needed. All right, next one. Okay, so those are the basic differences between DNA and RNA. So I hope you will never forget these differences, so that you will not be able to mix up these two nucleic acids that we have right here. All right, so let's see if you want to try this particular practice problem that we have right here. You can pause this particular video if you want to. All right, so now, in some cases, if you happen to see some books that contains this particular number 5 prime, and on the other side is 3 prime. All right, so what does the 5 prime and 3 prime means? It means that the 5 prime means that this end portion of the DNA, this one, is made up of a phosphate group. Yeah, this is the 5 prime. Alright, so this is made up of a phosphate group. Meanwhile, the 3 prime on this side is made up of hydroxyl group. So hydroxyl group. So basically that is OH. Alright, so meaning to say, if you form a structure using this particular sequence that we have right here, the first one at the beginning of this strand is made up of phosphate. It starts with phosphate and ends with the hydroxyl group. Now, what are we going to do here? We will find out the complementary strand that makes up this particular bases. So, as we all know, the base pairing rule, that the A is paired to T, then the C is paired to G, always. Alright, so let's start. We have here the A is paired to T, we have T is paired to A, then the C is G, then C. Next one, we have A over here, we have T, then on this letter we have G, we have C, we have A, we have T. Alright, so we have A again, we have C, we have G, we have G, we have T, and finally we have G. Alright, so did you get the correct answer? Alright, did you get the same answer as mine? Alright, so if you did get the same answer as mine, so meaning to say you did listen to what I've said on the lecture a while ago. Now, the problem that we have right here is, what will be the number? Alright, well, what will be the number that we will put here on this end that we have right here? So if this is the phosphate, so it should be 3 prime. Alright, so meaning to say that the hydroxyl group is on this side. Alright, so the hydroxyl group, so the OH, and on the other side, if this is the hydroxyl, it should have the 5 prime. Alright, so number on this side. So meaning to say that this side that you have right here, this is the phosphate side. Alright, so it contains the end part, which is the phosphate group. Alright, so again, 5 to 3, 3 to 5. Alright, so let's try this another set of strand. So if you want to try, you can pause this video and try it out first before you view the answer. Alright, so let's try. For example, we have here 3 prime on this side and 5 prime on the other side. So let's work this one out. So we have T, we have A, we have G, G, G. We have C, we have T, T, T. We have C, T, G, A. We have G, we have T, we have A, and finally we have T. Now, let's take a look at the number. So, if this is 3, it should be, on this side, it should be 5 prime. Now, if this is 5 prime, then should be, this is 3 prime. So, basically, what we did here today is the complementary strands that will be the basis for these strands that we have, or the given strands that we have right here. Alright, so basically, that's how you find the complementary strands of the DNA. Alright, so what we have right here, how about the RNA? Alright, so how about the RNA sequence that we have right here? For example, if we take, alright, so if we take this particular strand and try to find out its RNA equivalent, so we have A, T, T, T. So what will be its RNA strand? So we have U, we have A, and we have A. Alright, so basically this is your RNA strand. Now the only difference is in RNA we don't have T, instead we replace it by U. Alright, so basically that's how you find the complementary strands in DNA and the equivalent strands of DNA to RNA. Okay, so with that, in our next topic, we will try to find out how the DNA becomes, or how the DNA codes to become protein. Alright, so with the use of RNA, with the help of the RNA, how do you make proteins out of DNA blueprint that we have right here? Alright, so with that, today you have learned about the structure and function of the DNA and RNA. Alright, so also you have learned the base pairing rule, that the A is always paired to T and the C is always paired to G. And also you have learned that the Chargaff's rule, which states that there is always 1:1 ratio between the pyrimidines and purines in a particular DNA sequence in all organisms. And also, we studied the different types of RNA, and together with that is the differences between DNA and RNA. Alright, so with that ends up our discussion regarding the structure and function of the DNA and RNA, and I hope you did learn something from this video. Alright, so with that, special shout out to those to the sections that I'm handling. So we have 10 Ptolemy, 10 Poly, 10 Piano, and 10 Pythagoras. So thank you for the subscription on this particular video. So don't forget to comment down something on this video if you do have questions. So don't hesitate to write it down in the comment section. Alright, so with that, have a nice day everyone, and don't forget to smile. Alright, so goodbye and